to episode 6. Today I'll be discussing the lipid class of biomolecules and 3 of its subgroups: fats, steroids, and phospholipids. You probably know that the fat in our bodies and the butter we put on toast are lipids. Did you know that waxes, like beeswax or the wax in the leaf of a plant, are also lipids? Cholesterol and olive oil are lipids too. In this episode, I'll cover the structure of lipids, their qualities, their functions, and the role that lipids play in cells. I'll spend a portion of the episode talking about membranes, a naturally forming structure composed of lipid molecules, because the membrane is an extremely important aspect of the cell and life in general. The membrane is what defines the edges of the cell body. It regulates the in and outward flow of ions and various molecules. It separates and protects the cell's internal chemistry from the exterior environment. Membranes allow cells to concentrate useful chemicals where they can be easily accessed, instead of scrounging for the same chemicals free-floating in the solution. All of these things are critically important for the proper, stable functioning of living cells. Recall in episode 2, The Chemicals of Life, where I discussed hydrophobic and hydrophilic molecules. To briefly recap the relevant data, remember that hydrophilic molecules are those that have a polar or electronegative atom, like an oxygen in a hydroxyl group, or a nitrogen in an amino group. These molecules can interact with the polar water molecules, thus having the characteristic of being soluble in water. Hydrophobic molecules, on the other hand, have few, if any, of these electronegative atoms, and they don't interact very well with water molecules. When in an aqueous solution, hydrophobic molecules tend to clump together to minimize the surface area exposed to the water molecules. Due to their structure, lipids are hydrophobic. They cannot be dissolved in water or other polar solutions. The lipid class is very broad, defined as a carbon-containing compound that's both hydrophobic and nonpolar. These can be small chunks of carbon like isoprenes, free-floating chains of carbon like fatty acids, groups of chains like a triglyceride, or bulky heaps of carbon like steroids. Unlike proteins, nucleic acids, and carbohydrates, lipid molecules do not share a particular base of subunits to make polymers. Lipid structure is variable. Chain length, saturation, ring formation, all of these influence the structure and function of the lipid. Cholesterol, for example, is composed of 4 rings, 3 hexanes, and a pentane connected side by side with shared carbon atoms, with a 6-carbon chain with 2 methyl groups attached to the pentagon. This clump-like structure is characteristic of steroid molecules, which I'll discuss more in a few minutes. A simple fatty acid, on the other hand, is a little more than a chain of carbon atoms with a carboxyl group at the end. A carboxyl group has a carbon atom with a double bond to an oxygen atom and a single bond to another oxygen atom in a hydroxyl group. The carboxyl group has the chemical formula COOH, which can be spoken as Cu or Chu for easy reference. These fatty acid chains with their carboxyl ends can clump together to form various glycerides, like diglyceride or triglyceride. They can also bond to phosphate groups to become phospholipids, which are the primary molecule composing the membrane. I don't want to get ahead of myself and discuss the membrane in too much detail quite yet. For now, I'm going to stick with the structure of the most basic lipid, the simple fatty acid chain. When carbon atoms form chains connected by single bonds, the carbon atoms can rotate freely, which makes 3-dimensional bends in the chain. Think kind of like a thick piece of yarn being dangled around. When 2 of these carbon atoms share a double bond, the carbon atoms lose this freely rotating characteristic. If a double bond exists somewhere in a chain, it will produce a big kink in the structure. A double bond in a chain is almost like wrapping a piece of tape around a section of yarn. The piece of yarn is still flexible and can bend all over the place, except for that little segment covered with tape. That part's stiff and can't bend. Consider the structural diagram of a carbon chain that I described in the previous episode about carbohydrates. If you imagine a zigzagging line, each corner point represents a carbon atom, and each line segment represents a single bond between the carbon atoms. A double bond, however, can disrupt this nicely zigzagging line. Double bonds do not allow their atoms to rotate around each other like single bonds do. A double bond forces the carbon structure into a single plane. Try to imagine the following picture as I describe it, to get an image of how a double bond can affect the chain structure in a lipid like a fatty acid. First, imagine 2 dots, one above the other, connected with 2 parallel vertical lines. Each dot is a carbon atom, and the parallel lines represent the double bond. Imagine a horizontal line coming off the right side of the top dot, where it connects to another dot. This is the single bond and adjacent carbon atom further up the chain from the double bond. To continue the chain, imagine another line going up from this new atom, the bond to the next carbon atom. Then add another line going to the right, then another one up, then right, then up, then right again, and so on until you reach the carboxyl group at the end of this side of the carbon chain. Now let's come back down this zigzagging line of carbons, back to the double bond. The rest of this zigzagging chain is going to continue below the double bonded carbon atoms, but it can branch off either to the left or the right. Because the top of the chain is coming in from the top right, if the bottom of the chain continued to the bottom left, the chain itself would maintain a relatively linear structure. This configuration, where two groups branch off of different sides of a double bond, is called the trans configuration. The alternative configuration, where two groups branch off of the same side of the double bond, is called the Cysts configuration. If our hypothetical double bond had adjacent groups branching off on the same side, it would produce a kink. Imagine the zigzagging chain coming down from the upper right to the double bond, then branching down and to the right again. This creates an L shape that is responsible for the kink in the chain. When a fatty acid chain has no double bonds between any of its carbons, the chain has been saturated by hydrogen atoms. When a fatty acid chain has no double bonds between any of its carbons, the chain has been saturated by hydrogen atoms. Every carbon atom in the chain, with the exception of the carboxyl carbon and the other end-chain carbon, have two valence electrons shared with other carbon atoms and two valence electrons shared with hydrogen atoms. Basically, the saturated fatty acid chain is a polymer with CH2 monomers. The carboxyl carbon has 3 bonds to 2 oxygen atoms and a 4th bond with the first CH2 in the chain. The carbon at the other end of the chain simply replaces what would be the next carbon atom with a hydrogen atom. These fatty acids with no double bonds are called saturated fatty acids because the carbon backbone is saturated with hydrogen atoms. Fatty acid chains that have one or more double bonds are called unsaturated fatty acids because the chain isn't saturated with hydrogen atoms. If it were, hydrogen atoms would take the valence electron from each carbon involved in the double bond, and the chain would be saturated. Fatty acid chains with many double bonds are described as polyunsaturated. The saturation of a fatty acid molecule is fundamentally important to the macroscopic structure of the lipid. First of all, the length of the carbon chain is important. Short carbon chains do not offer much in the way of structural integrity, kind of like trying to make a blanket by tying a lot of really short pieces of string together. Longer carbon chains, on the other hand, can interact in a way not dissimilar from carbohydrates. Longer chains can overlap or pile together to form large sheets, which can be stiff and durable. Waxes, like beeswax, are composed of lipid molecules with very long saturated carbon chains. As a result, waxes like candle wax or the honeycombs of beeswax are stiff and solid at room temperature. Because of their linear structure, saturated carbon chains are easy to stack together tightly. This inherent ability to be packed densely contributes to the stiffness that is characteristic of waxes. Other saturated fatty acids that stay solid at room temperature exist in butter. Unsaturated and polyunsaturated fatty acids cannot pack tightly together as easily as saturated fatty acids can. The kink in the carbon chain is an irregularity that prevents the molecules from lining up side by side and crowding together, like they do in waxes. Because of this much looser organization between the lipid molecules, unsaturated fatty acids are usually liquids at room temperature. Olive oil, or safflower oil, or any other kind of organic oil substance are all composed of these unsaturated and polyunsaturated fatty acid chains. These oils are part of a subgroup of lipids called fats. The other biologically significant lipid subgroups are steroids and phospholipids. Fats are composed of molecular structures called triglycerides, which are themselves composed of 3 fatty acid chains and a glycerol molecule. Saturated fatty acids make solid fats, while unsaturated fatty acids make oils. The glycerol molecule is small, it's just a 3-carbon chain. But each carbon in this chain is also bound to a hydroxyl group, which means the glycerol molecule is polar, reactive, and soluble in water. A condensation reaction turns the oxygen in each hydroxyl group into an ester as it binds with the carboxyl group of the fatty acid chain. Steroids are heavier lipids characterized by a 4-ring structure. Earlier, I described the structure of the cholesterol molecule. The 3 hexane rings and the pentane ring are characteristic of all steroids, not just cholesterol. Steroids are differentiated by whatever side groups they happen to have attached to these backbone rings. One of the cholesterol's identifying features is the carbon chain attached to the pentane ring. Phospholipids are similar to fats, except one of the hydroxyl groups on the glycerol binds with a phosphate group instead of a third fatty acid chain. The phosphate group is also bound to a small organic molecule that carries a charge or some degree of polarity. This gives the phospholipid a few particularly interesting qualities. First, the phosphate group carries a strong negative charge, which means it's polar. The nonpolar fatty acid chains are repulsed by this polarity, which pushes the fatty acid chains to the opposite side of the glycerol. In diagrams, the phospholipid is typically drawn with the phosphate group on top and the fatty acid chains hanging off the bottom. This also means that the phospholipid has two distinct parts, the hydrophobic fatty acid chains and the hydrophilic phosphate group. This dual nature makes the phospholipid an amphipathic molecule, or a molecule with both hydrophilic and hydrophobic regions. Recall that all fatty acid chains are at least slightly amphipathic. The chains are hydrophobic, and the carboxyl group is hydrophilic. This amphipathic quality of phospholipids is extremely important, more so than any other quality that a lipid has. When placed in a water solution, amphipathic compounds do not dissolve. Instead, the hydrophilic heads of the phospholipid interact with water, while the hydrophobic fatty acid tails crowd together to avoid water. Because of this, groups of lipids like fatty acids or phospholipids form structures called micelles. A micelle is like a really small bubble. The outer surface of the bubble is a layer of carboxyl groups or phospholipid heads, the hydrophilic tips of the lipid molecules. The hydrophobic tails hang inwards from the surface of the micelle, the chains interacting inside the bubble. This micelle structure allows the hydrophilic regions to interact with water, while protecting the hydrophobic regions from coming into contact with water. Micelles are really small. There's little space left in the bubble that isn't filled with fatty acid chains. When collections of amphipathic molecules are large enough, they spontaneously form a structure slightly more complex than a micelle. When two sheets of phospholipids come together, they form what is called a phospholipid bilayer. To picture a cross-section of this bilayer, imagine two parallel lines of dots running horizontally. Each dot is the hydrophilic head of the phospholipid. Each dot in the top layer has two lines, the fatty acid tails, hanging down from it. These lines go about half the distance to the bottom line of dots. And each dot in the bottom layer also has two lines coming out of it, except these lines are going upwards towards the upper layer of dots, and they too go about halfway across the gap. At this point, you should be imagining two layers of phospholipids, their polar heads on the outside and their nonpolar tails on the inside. In this way, the hydrophilic surface of the bilayer protects the interior hydrophobic regions from water. The phospholipid bilayer protects its edges from water by wrapping itself into a sphere or bubble shape, with water both inside and outside the bubble membrane. Both micelles and bilayer membranes require no energy to form. They are simply the most stable resting states that large numbers of amphipathic particles take when they're dumped into a polar solution like water. The first cells used these bilayer membranes to isolate and protect their internal biochemical machinery from the outside world. Bilayer membranes give cells a surprising number of qualities and abilities that life depends on. Membranes provide a degree of permeability. They can shrink, expand, bud small bubble-like polyps, and host proteins, which are involved in all kinds of cellular activities. Membranes can also be used to create pressure or concentration gradients that can be exploited for energy, or to compartmentalize caustic chemical reactions where they won't hurt other parts of the cell. The permeability of the bilayer membrane refers to its capacity for allowing certain particles, like ions or small molecules, to pass through it. Consider two aqueous solutions separated by a bilayer membrane. A solute is put into one of the two solutions. If the membrane is permeable to that solute, the solute will begin to appear in the other solution, in the solution it was not originally poured into. The rate with which the solute enters the second solution can be used to calculate precisely how permeable the membrane is to that particular solute. This phenomenon, where some solutes pass through the membrane easier or faster than others, is called selective permeability. The bilayer membrane is most permeable to small, nonpolar molecules like CO2 and molecular oxygen, which can rapidly slide between the phospholipids. Despite their polarity, small and uncharged molecules like water can also leak through the membrane. These small molecules can pass through the membrane spontaneously via diffusion, or in water's case, osmosis. Larger molecules, or molecules with charges like ions, cannot easily move through the membrane without the active transport of specific proteins, which I'll discuss in more detail near the end of the episode. Not all membranes have the same permeability, however. Real quick, let me recap van der Waals forces, which were introduced in episode 3. Van der Waals forces are weakly polar interactions between atoms. They're created by the small polarity induced by the electron moving around the atom. The electron carries a negative polarity, while the opposite side of the atom carries a positive polarity. Membranes composed of phospholipids with kinked fatty acid tails cannot group together that tightly for the same reason that oils are liquid at room temperature. Because these phospholipids can't be packed as tightly, they aren't interacting as strongly through van der Waals forces between the carbon and hydrogen atoms in their fatty acid chains. This reduces the total cohesion and strength of the membrane. This kind of membrane is particularly permeable. Conversely, recall how waxes, like beeswax, are given stiffness and durability by their long fatty acid chains. Longer fatty acid chains can engage in more van der Waals interactions with one another, allowing them to be packed tightly together. Membranes composed of lipids with long fatty acid chains are thus less permeable to solutes. Cholesterols can also be part of a membrane. Because of their large, clumpy structure, the bulky rings of a steroid will close gaps by pushing the smaller phospholipids together. A simple membrane with short, unsaturated fatty acid tails is going to be far more permeable than a cholesterol-studded membrane with long, saturated fatty acid tails. Temperature of the membrane also plays a role in its permeability. At room temperature, a phospholipid membrane is like a liquid in its consistency. Imagine a swimming pool with its entire surface covered in floating bouncy balls, like the kind you'd find in a ball pit at the fair or an arcade. This layer of floating balls moves with the surface of the water like a membrane. An individual ball can move laterally along the surface of the water, just as phospholipids can move laterally within a layer of the membrane. At room temperature, phospholipids in a membrane move at about 2 micrometers per second. This means that really small cells, like bacterial cells, have membranes full of individual phospholipids moving the entire length of the cell in just 1 or 2 seconds. When the temperature rises, the movement speed of individual phospholipids, as well as the permeability of the membrane, increases. When the temperature drops, the movement speed slows and the permeability decreases. If the temperature drops enough, the membrane can freeze into a solid and lose all permeability. As the temperature drops, membranes with permeability-reducing features like kinked fatty acid chains and cholesterol molecules will lose their permeability faster than membranes with saturated chains and no cholesterol. Small, uncharged solutes can easily move across a membrane through a process called diffusion. I'm sure you know what diffusion means, but I'll explain it briefly just for the sake of clarity. All the particles are perpetually in a state of random movement. When you put a mass of particles into a solution, their random movement will cause them to slowly expand and fill the container, finding a level of equilibrium when the solution has a single, uniform concentration of the particle. Let's say that we have 2 solutions, again separated by a plasma membrane, and the same amount of 2 different solutes, one red and one blue. Both solutes are small, nonpolar particles that will diffuse easily across a membrane. One solution has the red solutes, while the other solution has the blue solutes. These solutes will diffuse across the membrane into the other solution because of a concentration gradient. The red solution, for example, is at a high concentration when put into the first solution. The second solution has no red solute, so the concentration is 0. Just as heat flows from hot items to cold items, solutes will exhibit a net flow from areas of high concentration to areas of low concentration. The red solute will thus diffuse across the membrane until both solutions have an equal concentration of red solute. The blue solute, meanwhile, is doing the exact same thing. Before the solutes fully diffused, one solution was colored red, while the other was colored blue. Both solutions appear purple because of the mixing of equal concentrations. The diffusion of water is called osmosis, and it occurs in membranes that are selectively impermeable to most or all other solutes. Imagine our 2 solutions separated by a membrane again. What if one solution had a higher concentration of solute than the other, but the solute could not pass through the membrane? There is still a concentration gradient at work here, but it isn't the solute's concentration gradient, it's water's. If you have 2 equal solutions, and put some mass of solute in one solution, that solution will have a higher concentration of the solute, but it will also have a lower concentration of water. Osmosis is then the net movement of water molecules going from areas of low solute concentration to areas of high solute concentration. This is cool and all, but what does it have to do with life? With cells? Recall that a bilayer membrane forms a bubble, or vesicle, with water on both the inside and outside. If water leaves the vesicle, it shrinks. If water comes into the vesicle, it swells. Imagine a cell floating in a solution with a constant concentration of some particular solute that is impermeable to the membrane. If the cell has the same concentration of solute inside its membrane as there is outside the membrane, there's no net movement of water molecules. The solution inside the cell is isotonic, as it shares the same solute concentration as the outer solution, and the shape of the cell is not changed by osmosis. Now imagine our cell has a very low concentration of solute within its membrane, making it hypotonic relative to the external solution. This is equivalent to saying the cell has a high internal concentration of water. Naturally, the water will leave the cell, and the cell will shrink. A shrinking membrane can look crumpled and wrinkled. Lastly, imagine the opposite case, where the cell has a very high concentration of the solute within its membrane, making it hypertonic relative to the external solution. Water from the exterior solution will come into the hypertonic cell, causing it to swell. Cells with a dangerously high solute concentration can swell so much that they burst, their membranes ripping and collapsing. This generally kills the cell, as the membrane collapse destroys all concentration and pressure gradients, and exposes the sensitive internal biomolecular machinery to the unregulated external environment. This capacity for membranes to swell or shrink plays an important role in numerous biological functions. Many plants, for example, have little pores in their external tissues called stoma, which are involved in the regulation of gas exchange. Two bean-shaped cells called guard cells border the stoma. When the plant needs to open the stoma, the guard cells become flooded with solutes, which makes their internal solutions hypertonic and attracts water through osmosis. The guard cells swell as they fill with water, but they can only extend lengthwise, as their width is constrained by rings of cellulose fibers. As the tips of each cell are connected, the swelling forces the cells to bow outwards, creating the opening of the pore between them. Gases then flow into or out of the pore, depending on the relative concentrations of internal and external gases. Remember when I mentioned the proteins that help move larger molecules and ions through the membrane? These proteins play a critically important role in the proper functioning of a cell's membrane, which is why I'm discussing them now and not in Episode 3. Speaking of Episode 3, remember how I discussed the side chains on the amino acids? Some of these side chains are nonpolar, while others are polar. When a contiguous sequence of amino acids has nonpolar side chains, that segment of the polypeptide is hydrophobic. Likewise, segments of the polypeptide with a large number of polar side chains are going to be hydrophilic. Just like the phospholipids, these proteins are amphipathic. When placed into a membrane, the hydrophobic region is attracted to the other hydrophobic fatty acid tails, where they can be stabilized by their own nonpolar interactions. By anchoring the protein in the membrane, the hydrophobic regions are protected from water, just like the fatty acid tails, and the protein is stabilized. The hydrophilic regions of the protein stick out on one or both sides of the membrane. Those that protrude from just the inner or outer surface of the membrane are called peripheral membrane proteins. Those that protrude from both sides of the membrane are called transmembrane or integral membrane proteins. Some peripheral proteins are only located on one side of the membrane, and most transmembrane proteins don't have similar structures on each side of the membrane. This means that the accessible protein environment on the inner surface of the cell is entirely different than the protein environment on its outer surface. This is an example of how membranes can isolate particular environments and compartmentalize reactions. Together, the various phospholipids and membrane proteins compose the cellular membrane in a kind of liquid mosaic. If you would like to read more about this, look up the fluid mosaic model, which is the current working model for the overall composition and organization of the membrane. So what are these proteins actually doing in the membrane? Some of these proteins, like glycolipids, are involved in cell signaling and identification. Others are involved in physically stitching cells together to form tissues. To stay on topic, I'm going to be discussing the membrane proteins that affect permeability. Of these, there are three main groups, each with a very wide variety of structures. These groups of proteins, in order of increasing complexity, are called channels, carriers, and pumps. Channel proteins are selective, tube-like structures that allow a specific ion or molecule to move across the membrane. The interior lining of an ion channel is often charged or polarized to attract the specific ion that it shuttles across the membrane. Channels that move water molecules are called aquaporins, which literally means water pore. There are two qualities in aquaporins that allow it to selectively transport water molecules. The first is the polarity of the interior lining of the channel, which helps pull water molecules along. The second quality is a number of specific amino acids in the protein structure that physically blocks larger molecules or ions from entering. There are gated channels, which open and close in response to a particular chemical stimulus. Voltage-gated channels, for example, only respond if the membrane is depolarized. Ligand-gated channels only respond if they bind to a specific ligand, like a sodium ion, a neurotransmitter, or a hormone. Channel proteins require no energy input to work. As small nonpolar ions can diffuse across the membrane through passive transport, channels enable ions and larger particles to move across the membrane through facilitated diffusion. Carrier proteins, the second group, are very similar to channel proteins. They're tube-like proteins that provide a route for specific particles to cross the membrane, while requiring virtually no energy input. Carriers differentiate themselves from channels by the manner of their transport. Carriers are like gated channels, except the item they must bind to open is the same molecule that they transport across the membrane. A literal textbook example is the GLUT1 transporter. All cells use glucose, either to store energy or for structural purposes. Unfortunately for cells, glucose is pretty large and dotted with polar hydroxyl groups, so it has a real hard time diffusing through the membrane. The GLUT1 transport protein is like a tube with its middle squeezed closed. Outside the cell, a glucose molecule is attracted to the GLUT1 protein, where it binds to the protein's active site. Once bound, the GLUT1 protein begins to bend and contort. Its shape flexes in response to the bound ligand, the glucose molecule. As the GLUT1 transporter is altering its shape, the glucose molecule is dragged through and released on the inside of the membrane. After bringing a particle into the cell, the transport protein will naturally reverse its shape after it's let go of the particle, or it will wait to bind another particle inside the cell, which it then transports to the outside. Protein pumps are the third group, capable of moving ions or molecules against their concentration or electrochemical gradients. Unlike channels and carriers, moving particles against the various gradients that affect them takes energy. Protein pumps consume ATP, a molecule I described last episode, for the energy they need to work. The third phosphate group breaks off of the ATP and binds to the pump, giving it the potential energy it needs to change shape, in addition to the polar interactions it has with the protein side chains. Some pumps move hydrogen ions, others move calcium ions. Probably the most famous protein pump of all time is the sodium-potassium pump, which helps the cell breathe in potassium ions and breathe out sodium ions. On the inside of the pump, there are 3 active sites with a strong affinity for sodium. When 3 sodium ions bind to the pump, the ATP deposits a phosphate group. This makes the pump change shape. A pore is opened, and the binding sites suffer a reduced affinity for the sodium ions. The ions detach and float out of the cell. The now empty pump is exposed to the outside. It also has 2 new binding sites with a strong affinity for potassium. 2 potassium ions outside the cell then bind to the pump, just like the calcium ions did on the other side. When the potassium ions bind, the phosphate group is detached from the pump. The shape-altering effects of the phosphate group also disappear, and the pump returns to its original conformation. This just so happens to expose the potassium ions to the inside of the cell, and reduces the binding site affinity for potassium. The pump is now back where it started, and it can transport more sodium ions out of the cell. In this way, it pumps sodium out and potassium in. Protein pumps of all sorts can be used to maintain or create electrochemical or concentration gradients, and these gradients can themselves be exploited as a source of energy. Co-transport occurs when one pump sets up a gradient, and this gradient provides the energy for one or more different pumps to function. Okay, that was a long episode about lipids, but I think I covered most of what I needed to cover. Let me recap. Lipids are largely hydrophobic, carbon-rich molecules. In aqueous solutions, some lipids can spontaneously form micelles and bilayer membranes. These bilayer membranes would naturally form bubbles, which were then worn by self-replicating molecular machinery like a cloak to make the first cells. Cells use these bilayer membranes to create isolated chemical environments. They isolate their internal chemistry from the outside environment, and they internally isolate caustic reactions in smaller internal membrane bubbles, or endomembrane bubbles. The variety of functions expressed by the membrane proteins allows the cell to maintain these isolated chemical environments, which in turn keeps the cell alive. Well, that's about it for lipids. This is also the final episode in the first mini-series of the podcast. Episodes 1 through 6 covered the biochemistry of life. Episode 2 was an introduction to the basics of chemistry, and episodes 3, 4, and 5 covered proteins, nucleic acids, and carbohydrates respectively. This episode covered lipids, the final major class of molecules that compose all living beings. If you liked this mini-series, you can get excited because I plan to make more. In the future, I plan on making mini-series about, among other things, the evolution of life across eons of geologic time, the various systems of the human body, and the 5 major classes of vertebrates. I hope that from this podcast you were able to learn something cool. As always, thanks for listening. If you enjoy the podcast, tell your friends, tell your teachers, tell your classmates. Hit the like button and subscribe so you can see new episodes when they release. The BioLogic podcast is kept alive by the support of listeners like you. If you want to help support the podcast, please consider buying some merchandise in our official store or becoming a patron by visiting our Patreon page. The official BioLogic store can be found at redbubble.com/people/biologic/shop. In the store, you can buy merchandise like t-shirts and coffee mugs and hoodies. All the products have really cool biology-themed artwork on them that I've designed myself. The official BioLogic Patreon page can be found at patreon.com/biologicpodcast. The support of Patreon patrons is very important for the show. Longtime patrons are rewarded with exclusive episodes on fascinating topics and will have their questions prioritized in AMA episodes. Thanks for your support, and as always, thank you for listening.